Good morning, students. Today we'll be doing the experiment determination of biochemical oxygen demand BOD for a given sample of water. So we'll be determining the total quantity of BOD in a given sample of water or wastewater, whatever is the case. Okay. So I think before we discuss the BOD, you must have a clear-cut idea of the dissolved oxygen, how to determine the quantity of dissolved oxygen in a given sample of water, because that is a prerequisite for determination of BOD. I think that experiment has already been conducted online, okay, by one of our faculties. So dissolved oxygen is a prerequisite for biochemical oxygen demand. Why? Because BOD is simply given as the difference between initial BOD and initial DO and final DO multiplied by the dilution ratio. Okay, you know dissolved oxygen is present normally in water, and with time the dissolved oxygen level depletes. With the passage of time, the dissolved oxygen level keeps on decreasing. Okay, so as the time goes on, okay, the dissolved oxygen level decreases. So basically initial DO is always going to be higher with respect to, so say 4-day dissolved oxygen or 5-day level of dissolved oxygen. In same water sample which was having an initial amount of DO, after 5 days if you will check, will be noticing that the DO level has depleted. Why? Because there are microorganisms, aerobic bacteria present in the water sample, and they consume the dissolved oxygen for oxidizing the organic matter that is present in the water. And more specifically in wastewater, high quantity of organic matter is present. Okay, so we will discuss in details how do we determine the BOD and what will be the procedure, means how we will proceed for the lab. We will first discuss the theory and principle involved in the biochemical oxygen demand BOD test. Then we will be learning about the apparatus and reagents required. Then I will be telling you the procedure involved. Then I will be showing you the lab video that we have prepared for you, how to determine the BOD. Then I will be telling you the calculation and conclusion, and as well as the environmental significance of this biochemical oxygen demand. So basically students, biochemical oxygen demand, you know, it is the total quantity of oxygen required for basically oxidizing the organic matter present in the wastewater, okay, by the aerobic bacteria and other microorganisms that require oxygen. So it has been written, biochemical oxygen demand is the amount of dissolved oxygen needed by aerobic biological organisms to break down the organic material present in a given water sample at a certain temperature over a specific time period. Okay, the biochemical oxygen demand is an empirical standardized laboratory test, okay, which measures oxygen requirement for aerobic oxidation of decomposable organic matter. You know students, there are always organic matter present in the wastewater, okay, even in water sample, and they keep on being oxidized or decomposed by the aerobic bacteria and other microorganisms that are present in the water, and in turn the dissolved oxygen level of the water or wastewater keeps on decreasing. I have told you also that a minimum 4 mg per liter of dissolved oxygen should be there in water at any time, otherwise fishes and other aquatic organisms might get killed. Okay, so a certain amount of dissolved oxygen, minimum more than 4 mg per liter, should always be there, and a healthy dissolved oxygen level would be 10 to 20 mg per liter of dissolved oxygen. Okay, so the biochemical oxygen demand is an empirical standardized laboratory test which measures oxygen requirement for aerobic oxidation of decomposable organic matter and certain inorganic materials in water, polluted waters and wastewater under controlled conditions of temperature and incubation period. You know BOD is calculated as 20 degree centigrade incubation for 5 days, है ना. हम क्या करते हैं, जिस वाटर सैंपल में हमें BOD डिटरमाइन करना है, वी स्टोर इट इन इंक्यूबेटर. इंक्यूबेटर इज से अ टाइप ऑफ टेम्परेचर कंट्रोल डिवाइस. जैसे फ्रिज में हम जो है, कुछ फ्रिज में हम टेम्परेचर मेंटेन कर सकते हैं ना, तो आप उसको ऐसा ही समझ लीजिए कि इट्स अ टेम्परेचर कंट्रोल डिवाइस वेयर अ पर्टिकुलर टेम्परेचर कैन बी मेंटेन्ड कंटिन्यूसली एंड हेक्टिकली. ओके, से 5 days, for 5 days we set temperature as 20 degree centigrade. We take water sample, we determine the initial DO level, dissolved oxygen level, then we store the water sample for 5 days at 20 degrees centigrade in the incubator. Okay, and after 5 days we take out the water sample, we determine the final dissolved oxygen level, and then we can easily calculate the BOD. BOD is given as the initial DO minus final DO. Okay, that difference multiplied by the dilution ratio. What is this dilution ratio? Dilution ratio is basically the ratio of volume of diluted sample divided by volume of undiluted sample. For example, say we are taking 10 milliliter water sample. हम कितना ले रहे हैं? 10 milliliter water sample ले रहे हैं, and we are adding distilled water into the water sample so that we increase the volumes to 100. तो हमने क्या किया? 10 मिलीलीटर वाटर सैंपल लिया. अनडाइल्यूटेड कितना है? 10 मिलीलीटर. उसके बाद हम क्या करते हैं स्टूडेंट्स? वी डू अ वॉल्यूम मेकअप बाय एडिंग डिस्टिल्ड वाटर एंड वी प्रिपेयर 100 मिलीलीटर डाइल्यूटेड वाटर सैंपल. हमने क्या किया? 100 मिलीलीटर जो है उसका वॉल्यूम बढ़ा दिया डिस्टिल्ड वाटर ऐड करके. तो व्हाट विल बी द डाइल्यूशन रेशियो? इट विल बी वॉल्यूम ऑफ डाइल्यूटेड सैंपल बाय अनडाइल्यूटेड सैंपल, दैट इज 100 डिवाइडेड बाय 10. सो द डाइल्यूशन रेशियो वुड बी 10. ठीक है ना? वॉल्यूम ऑफ डाइल्यूटेड बाय अनडाइल्यूटेड सैंपल. सो BOD सिंपली गिवन एज the DO difference multiplied by the dilution ratio. Okay. So the quantity of oxygen required for above oxidation process is a measure of the test. The test is applied for fresh water sources like rivers, lakes. You know, rivers and lakes are having ample quantity of dissolved oxygen, and they are having the capacity to decompose, oxidize huge quantity of organic matter. Okay, and that is why the DO level would also be high. Wastewater, that is domestic and industrial wastewater, polluted receiving water bodies, marine water, that is estuaries, coastal water. Coastal water are the water available in sea and oceans. Okay, all those regions they contain coastal water. Estuaries are basically the point where a river enters basically a sea, है ना? जिस पॉइंट पे रिवर जो है सी में एंटर होता है, उस पॉइंट पर आप देखिएगा कि देयर आर डेल्टाज एंड एस्टुअरीज फॉर्म. एस्टुअरीज कंटेन स्वीट वाटर एंड दे आर वेरी गुड ब्रीडिंग ग्राउंड फॉर फिशेस, है ना? वहाँ पे मच्छी का जो है वहाँ पे प्रोडक्शन बहुत अच्छा होता है. एस्टुअरीज आर वेरी गुड फॉर कल्टिवेशन ऑफ फिशेस. फिशी कल्चर के लिए बहुत अच्छा होता है. Polluted receiving water bodies, marine water, estuaries and coastal water, and also for finding out the level of pollution, assimilative capacity of water body, and also the performance of wastewater treatment plants. Now BOD reduction is used as a gauge of the effectiveness of water treatment plants. BOD of wastewater effluents is used to indicate the short-term impact on the oxygen levels of receiving water. BOD analysis is similar in function to chemical oxygen demand analysis, in that both measure the amount of organic compounds in water. तो हम क्या करते हैं? बेसिकली BOD reduction is used as a gauge or as a measurement of the effectiveness of water treatment plants. Water treatment plant कितना effective है? That depends on the capacity of that water treatment plant in reducing, in decreasing the BOD level. Okay, BOD of wastewater effluents is used to indicate the short-term impact on the oxygen levels of receiving water. पानी में जो है अगर BOD हाई है, इसका मतलब वो बहुत ज़्यादा पोल्यूटेड है, and that will consume huge quantity of dissolved oxygen. So the aquatic organisms are going to be in threat if the BOD level are high. Okay. BOD analysis is similar in function to chemical oxygen demand. You know, one more oxygen demand is there, chemical oxygen demand, COD, where aerobic, where organic matter as well as inorganic matter are oxidized chemically. Okay, there we add a strong oxidizing agent like potassium dichromate, and that increases the dissolved oxygen level, and that is why the not only dissolved oxygen level, that helps in oxidizing the matters, organic matter as well as inorganic matter. Okay, in both measure the amount of organic compounds in water. So our objective is determination of biochemical oxygen demand BOD for a given sample of water, and I have told you students that dissolved oxygen DO is a prerequisite for biochemical oxygen demand, because we measure initially the dissolved oxygen level in the water sample. then we incubate the water sample after diluting it. First we measure the initial DO level, okay, then we dilute the water sample, okay, and we store it for 5 days, and then we determine the final oxygen level, okay, and that difference between initial oxygen level and initial dissolved oxygen and final dissolved oxygen multiplied by the dilution ratio gives us the BOD for 5 days at 20 degrees centigrade. Okay, so what are the apparatus and reagents that are involved in the experiment? Basically we will need a BOD bottle, okay, we will need a BOD bottle of 300 ml कैपेसिटी. BOD बॉटल जो है नॉर्मल जो हमारा फ्लास्क वगैरह होता है उसी तरफ का देखने में. I'll show you in the experiment in the video. So BOD bottle has a capacity of 300 milliliter. 300 milliliter जो है उसका capacity है. We clean it properly with detergent, rinse thoroughly and drain before use with water seal. Okay. So basically BOD bottle 300 ml का वो हमें चाहिए. We need an incubator. Incubator I've told you it's a temperature controlling device. Looks similar like a refrigerator. Refrigerator या फ्रिज वगैरह उसी तरह देखने में होता है incubator, and it is used for basically maintaining a particular temperature level. Okay, तो क्या करता है उसमें particular temperature level जो है maintain कर देता है. Say 20 degree centigrade maintain कर सकता है. We can maintain 27 degree centigrade and it can work continuously for 2 days, 3 days, 5 days. Okay, we will also require burettes, pipettes, है ना. हमें burette की भी जरूरत पड़ेगी because in the burette we will do titration. Okay, then we will need pipette because we have to pipette fixed required quantity of reagents, right, in the to add in the water sample. And also we will require the following chemical reagents. Okay, we will require manganese sulfate. Okay, we will require alkali iodide azide. We will require concentrated H2SO4. We will need starch as an indicator, and we will need standard sodium thiosulfate. Okay, we will need standard sodium thiosulfate of 0.025 normality. You will see, students, that all the reagents, okay, that is required in the experiment, they are basically same which are required in dissolved oxygen. DO का जो हमने लैब किया था उसमें जो है जितने भी reagents required थे, you will see that more or less same ingredients, same reagents are required in determination of BOD, because BOD is eventually that difference between initial DO and final DO. Initially कितना dissolved oxygen है, finally कितना dissolved oxygen, उसका difference just multiplied by dilution ratio, which is the volume of diluted sample divided by volume of undiluted sample, that is the dilution ratio. ठीक है ना. इसीलिए आप देखिएगा कि जितना भी ingredients है या reagents है, chemical reagents, they are same as the dissolved oxygen. Okay. Now I will tell you the procedure and then I will show you the lab video that we have prepared for the BOD experiment. And the lab video will be basically determination of dissolved oxygen only. We will determine initially the dissolved oxygen level, then after 5 days we determine the final dissolved oxygen level, and then just we multiply it with the dilution ratio and we get the quantity of BOD that is present in the water sample. So first we collect sample in a BOD bottle, okay, using DO sampler. Then we add 1 milliliter of MnSO4, okay, that manganese sulfate. Manganese sulfate we add 1 milliliter, followed by 1 milliliter of alkali iodide azide reagent to a sample collected in 200 to 300 milliliter bottle. Basically हम क्या करते हैं students, we take the BOD bottle, right, we add 1 milliliter of MnSO4, we add 1 milliliter of alkali iodide azide. Okay, the tip of the pipette should be below the liquid level while adding these reagents. Then a stopper is put immediately and pipette is rinsed before putting them to reagent bottles. उसके बाद क्या करते हैं students, after having added 1 milliliter of MnSO4 and 1 milliliter of alkali iodide azide, right, the bottle is given little bit movement. हम जो है थोड़ा सा उसको जो है shake करेंगे bottle को, okay, so that proper mixing takes place. And we will see a brownish color being developed over there. हम देखेंगे कि एक brown color का जो है color develop हो रहा है. Then we will let it लेट द बॉटल रिमेन फॉर से 30 to 40 मिनट्स, 30-40 मिनट तक जो है बॉटल को अनडिस्टर्ब्ड कंडीशन में रख देंगे. ओके, देन वी विल सी दैट द brown color that was being formed that has turned into a precipitate and has settled down at the bottom. ठीक है, प्रेसिपिटेट क्या होता है? जब कोई सॉलिड जो है जो कि लिक्विड में डिजोल्व नहीं हो रहा है वो सेटल डाउन हो जाता है एट द बॉटम. डोंट गेट कंफ्यूज्ड विद हाइड्रोलॉजी प्रेसिपिटेशन. ओके, वो उसकी बात नहीं कर रहा हूँ मैं. इन केमिस्ट्री एंड एनवायरमेंट लैब whenever you will hear the word precipitate in liquid, that means some solid is being talked about which has not dissolved into the liquid but has settled down at the bottom. कोई insoluble solid जो कि बॉटम में सेटल कर जाए उसको हम कहते हैं precipitate. तो मैंने आपको क्या बोला, we take BOD bottle, we add 300 बेसिकली BOD बॉटल लेते हैं हम, ठीक है ना. we take the water sample, ठीक, we take water sample and we dilute it up to say 300 ml because the capacity of the BOD bottle is 300 milliliter. हम क्या करते हैं, सपोज हमने 50 milliliter water sample लिया, suppose we have taken 50 milliliter water sample and we dilute it in the BOD bottle which has a capacity of 300 milliliter. हम 50 ml को डाइल्यूट करके 300 ml जो है वॉल्यूम बना देते हैं. कैसे डाइल्यूट करते हैं? डिस्टिल वाटर ऐड करके. ठीक है, तो हमने क्या किया, 300 ml जो है बेसिकली हमने वॉल्यूम बना दिया BOD बॉटल में. ओके, 50 ml वाटर सैंपल लिया, उसमें हमने डिस्टिल वाटर ऐड करके 300 जो है वॉल्यूम्स का बना दिया. तो व्हाट विल बी द डाइल्यूशन रेशियो? डाइल्यूशन रेशियो विल बी बेसिकली वॉल्यूम ऑफ डाइल्यूटेड सैंपल बाय वॉल्यूम ऑफ अनडाइल्यूटेड सैंपल. व्हाट इज द वॉल्यूम ऑफ डाइल्यूटेड सैंपल? 300 मिलीलीटर, है ना, क्योंकि हमने 300 मिलीलीटर बना दिया ना वॉल्यूम. एंड व्हाट इज द volume of undiluted sample? That was 50 ml that we had taken. So what will be the dilution ratio? It will be simply volume of diluted sample by volume of undiluted sample. Volume of diluted sample 300 है, undiluted sample का volume 50 है. So 300 divided by 50, that means 6. Dilution ratio is 6. Then in the 300 milliliter diluted sample we add 1 milliliter MnSO4 excuse me, and 1 milliliter of alkali iodide azide. हम जो है 1 milliliter MnSO4 ऐड करते हैं, 1 milliliter जो है alkali iodide azide उसमें ऐड कर देते हैं. अब brownish color will develop. We will let it remain for 30 to 40 minutes. 30-40 मिनट तक उसको हम जो है undisturbed condition में छोड़ देते हैं. And we close the cap also. बोतल बंद करके छोड़ देते हैं अच्छे से एक बार मिक्स करने के बाद. We will see that within 30 to 40 minutes there is a clear cut precipitate that has settled down at the bottom. Brown precipitate जो है वो बॉटम में जो है बोतल के, है ना, बोतल के नीचे आप देखेगा एक ब्राउन कलर का precipitate जो है सेटल डाउन हो गया. ठीक, एंड देयर इज अ क्लियर लिक्विड above the top of the precipitate. उसी बोतल में ऊपर हम देखेंगे कि एक क्लियर लिक्विड लेयर है, उसको हम सुपरनेटेंट भी बोलते हैं. सुपरनेटेंट क्या होता है? इट इज द लिक्विड लेयर लाइंग अबव द इनसॉल्युबल सॉलिड प्रेसिपिटेट. ठीक है, हम देखें कि क्लियर लिक्विड है जो कि ऊपर में दिख रहा है और नीचे में, मैं अभी आपको जब एक्सपेरिमेंट दिखाऊंगा तो समझ में आएगा. एंड नीचे में जो है इनसॉल्युबल सॉलिड जो है सेटल डाउन हो गया. ओके, दैट इनसॉल्युबल सॉलिड इज ड्यू टू ब्राउन प्रेसिपिटेट ऑफ MnO2 दैट इज फॉर्म. ठीक है, देन व्हाट वी डू, वी विल when we see that clear supernatant is formed, यानी क्लियर लिक्विड लेयर जो है टॉप में दिख रहा है, नीचे जो है प्रेसिपिटेट डिपॉजिट हो चुका है. द प्रेसिपिटेट इज वाइट इफ द सैंपल इज डिवॉइड ऑफ ऑक्सीजन. तो ऐसा हो सकता है कभी कभी वो प्रेसिपिटेट जो है व्हाइट कलर का दिखे, इसका मतलब डिजॉल्व ऑक्सीजन लेवल बहुत कम है. अदरवाइज इट विल बी ब्राउन इन कलर. ओके, नाउ वी एड 1 milliliter ऑफ कंसनट्रेटेड H2SO4. उसके बाद हम क्या करते हैं स्टूडेंट्स, वी विल एड 1 milliliter ऑफ कंसनट्रेटेड H2SO4. व्हेन आई से कंसनट्रेटेड, इसका मतलब स्ट्रॉन्ग एसिड है. आप जानते हैं ना, कंसनट्रेटेड और डाइल्यूटेड दो कैटेगरीज होता है. इफ इट इज कंसनट्रेटेड इट्स अ स्ट्रॉन्ग वन. इफ इट इज डाइल्यूटेड दैट मीन्स द स्ट्रेंथ हैज बीन रिड्यूस्ड बाय डाइल्यूटिंग इट विद डाइल्यूटिंग एजेंट्स. ठीक है ना. So we need to handle the acid very cautiously because it's a strong acid. Okay, it's a concentrated acid. So we add in the BOD bottle where a brown or a whitish precipitate has settled down at the bottom and a liquid layer is at the top. We add 1 milliliter of concentrated H2SO4 very carefully. Okay, Then we replace the stopper and mix well till the precipitate goes into the solution. As soon as we add 1 milliliter of concentrated H2SO4, the entire chemical and basically precipitate and the liquid they mix properly and the precipitate disappears. जो भी ब्राउनिश कलर का या वाइट कलर का जो प्रेसिपिटेट था आप देखेगा वो पूरा घुल गया, ठीक है, इट डिजॉल्व्स entirely. Then what we do is we will take 201 milliliter और समटाइम्स आप देखिएगा 100 से 150 या 250 तक भी जो है we take water from the BOD bottle. Okay, here it has been written precisely 201 milliliter. We can take, otherwise we will take 100 to 150 ml of water sample from the BOD bottle in a conical flask. हमें conical flask में जो है 150 से 250 या फिर 201 ml भी ले सकते हैं of the solution we take, okay, in a conical flask and we add starch as indicator. हम क्या करेंगे, ये जो BOD बॉटल है जिसमें कंसनट्रेटेड H2SO4 ऐड कर दिया गया है वहाँ से हम जो है 100 से 150, 100 to 150 और sometimes we add 201 milliliter of the bottle to be precise in a flask and we add starch as indicator. ठीक, जैसे ही हम उस बोतल में स्टार्च इंडिकेटर, जैसे ही फ्लास्क में हम BOD सैंपल ऐड करेंगे और स्टार्च इंडिकेटर ऐड करेंगे, इट विल टर्न ब्लू इन कलर और ब्लूइश ग्रीन इन कलर. ठीक है, कॉनिकल फ्लास्क में हमने BOD सैंपल लिया, वी हैव एडेड स्टार्च एज इंडिकेटर, इट विल आइदर टर्न ब्लू इन कलर और ब्लूइश ग्रीन इन कलर. ओके, देन वी विल टाइट्रेट इट अगेंस्ट स्टैंडर्ड सोडियम थायोसल्फेट सोल्यूशन. सोडियम थायोसल्फेट सोल्यूशन जो है 0.025 नॉर्मेलिटी का, ओके, 0.025 नॉर्मेलिटी का सोडियम थायोसल्फेट सोल्यूशन है, दैट वी ऐड इन द ब्यूरेट एंड वी टाइट्रेट द BOD sample taken in the flask, okay, where starch was added as an indicator. We will titrate it against the standard sodium thiosulfate solution in the burette. So initial what is the color of the sample in the conical flask? It is either blue in color or bluish green in color. Then after titration, when it has been titrated against standard sodium thiosulfate solution of 0.025 normality, the end point will be basically colorless. वो blue color से या फिर bluish green color से the water sample in the flask will turn colorless. जैसे colorless हो गया, we will note down the final volume. We will note down the initial volume of sodium thiosulfate. Then as soon as the water sample in the conical flask turns colorless, we will note down the final volume. So what is the volume of sodium thiosulfate that is consumed? Final volume minus initial volume is the volume of sodium thiosulfate that is consumed. Okay, and then we will do calculation to find out the initial DO. Initial DO find out करने के बाद we will store basically हमने जो है वाटर लेवल डिटरमाइन, DO लेवल डिटरमाइन कर लिया ना, जो वाटर सैंपल है उसमें इनिशियल DO कितना हमने डिटरमाइन कर लिया. ठीक, देन वी विल टेक द वाटर सैंपल, ओके, वी विल स्टोर इट in the incubator for 5 days at 20 degree centigrade. Then the water sample is kept in 300 milliliter BOD bottle for 5 days at 20 degree centigrade as an incubator. हमने क्या किया स्टूडेंट्स, हमने जो है BOD लेवल इनिशियली, DO लेवल, आई एम सॉरी, आई बेग योर पार्डन. सो हमने DO लेवल इनिशियली जो है डिटरमाइन कर लिया वाटर सैंपल में. ठीक है, कैसे, ये एंटायर प्रोसीजर हमने डिस्कस किया उससे. देन वी टेक BOD बॉटल, वी ऐड 300 मिलीलीटर सैंपल. हमने इनिशियली डाइल्यूट किया था क्योंकि वी नीडेड टू नो बेसिकली द डाइल्यूशन रेशियो ऑल्सो. देन द वाटर सैंपल इज केप्ट इन द 300 ml BOD बॉटल फॉर 5 days at 20 degree centigrade inside the incubator. After 5 days are over, the sample is taken out. Okay, the final DO is determined. How do we determine the final DO? Similarly, same procedure से जैसे हमने इनिशियल DO डिटरमाइन किया उसी तरह से जो है हम जो है फाइनल DO भी डिटरमाइन करेंगे. उसमें भी फिर एंटायर प्रोसीजर विल बी रिपीटेड. ओके, सो द फाइनल DO लेवल इज डिटरमाइंड इन द वाटर सैंपल आफ्टर 5 days at 20 degree centigrade. Incubation period is over and then we find out the BOD by the formula final DO, initial DO minus final DO, because initial DO is going to be more than final DO. इनिशियली DO ज़्यादा होता है, फिर DO घटने लगता है. 5 दिन के बाद जो है DO लेवल काफ़ी कम हो जाता है. सो that initial DO, which is higher one, minus final DO gives us the quantity of DO that is consumed in oxidizing the organic matter. Quantity of DO that is consumed in oxidizing the organic matter. So initial DO minus final DO into the dilution ratio. What was the dilution ratio? Volume of diluted sample by volume of undiluted sample. That is 300 milliliter divided by 50 milliliter. Say, ठीक है ना? That will give us the BOD after 5 days at 20 degree centigrade. This is the procedure explained for determination of DO. Okay, we have taken BOD bottle. We have added, okay, 1 milliliter of manganese sulfate, 1 milliliter of alkali iodide azide. Then we have given a little bit of shaking. Okay, then brownish precipitate forms at the bottom and a clear liquid layer at the top of the bottle. Then we add 1 milliliter of concentrated H2SO4, right, and we shake it a little bit so that the entire precipitate gets dissolved. Then we take 201 milliliter of sample in the flask, or we can take 100 to 150 milliliter of water from this BOD bottle into the flask, and we add starch as indicator. Blue color or bluish green color is developed. Then we titrate it against standard sodium thiosulfate solution of 0.025 normality. Then the color changes from blue or bluish green to colorless. Okay, and we note down the final volume, and then we calculate the DO. Then we store the bottle for 5 days at 20 degree centigrade. After 5 days we take out the bottle, we determine the final DO, and then we find out BOD by formula DO consumed, that is initial DO minus final DO into the dilution ratio. As simple as that. Okay, now I'll show you the basically lab video that we have prepared for the purpose. Okay, so here you see water sample is there. Okay, this is the BOD bottle, students. This is the 300 ml BOD bottle. Okay, this is manganese sulfate. This is alkali iodide azide. 1 milliliter of when we will take water sample in this. Okay, we will take 50 milliliter of water sample initially. We will do volume makeup up to say 300. Then we will add 1 milliliter manganese sulfate, 1 milliliter of alkali iodide azide. Okay, then we will mix it properly. A brown precipitate will be formed and a supernatant liquid layer will be formed at the top of BOD bottle in this portion. Okay, then we will add 1 milliliter of concentrated H2SO4. Entire precipitate will get dissolved. Then we will take 201 milliliter, or we can take 100 to 150 milliliter also, doesn't matter, of the water sample in a flask. Okay, and we will add starch indicator, and we will titrate it against standard sodium thiosulfate solution. Okay, so basically you are seeing the BOD bottle, manganese sulfate, then alkali iodide azide, water sample is taken. Okay, this is the standard sodium thiosulfate solution. This is the starch indicator. This is concentrated H2SO4. These are the pipettes. Here you see the pipettes. Now this BOD bottle, okay, 300 ml capacity. This is the stopper. This is the stopper or the cork. Water sample is taken. Okay, the water sample is taken. This is complete 300 milliliter. We can also take suppose 50 ml water sample and do volume makeup up to 300, and we should do that for finding out the dilution ratio. Okay, for finding out the dilution ratio we have to dilute. Now this is the pipette. We will take 1 ml of magnesium sulfate and 1 ml of alkali iodide azide. These are the markings. This is 10 milliliter pipette. 1 milliliter marking at each point is there. Okay, 1, 2, 3, 4 like that 10. We are taking 1 milliliter of MnSO4, magnesium sulfate. We are taking 1 milliliter. We will add into the BOD bottle sample. This is the burette. Okay, this is the burette stand. We can adjust it so that the flask can be fitted at the bottom without any obstruction. So, these are the sodium thiosulfate will be put into this burette. Okay, this is the way to stop and start the flow of liquid in the burette. This is on, this is off. This is on. Okay, liquid will flow. This is off, liquid won't flow. Sodium thiosulfate will be put into this burette. Now in the BOD bottle we will add 1 milliliter MnSO4 magnesium sulfate. We will add 1 milliliter. Very carefully we have to take out 1 milliliter magnesium sulfate solution. Okay. Not more than that. This is 1 milliliter. In the marking you see 7 to 8 means we will release 7 to 8. Okay, that will be 1 milliliter, and remaining we will put into the. This is alkali iodide azide. 1 milliliter we will take. Now you will see that a brown precipitate will be forming. Initially a total yellowish brown color will develop, and then we will leave it for 30 minutes, 30 to 40 minutes, and we will see that a precipitate, brownish precipitate or yellowish brown precipitate is formed, which deposits at the bottom, and a clear supernatant liquid is formed at the top of this BOD bottle. Okay. We have mixed it properly, then we will leave it for 30 minutes, and we will see a brownish precipitate. 30 minutes period is over. You see students, a precipitate is formed at the bottom. A precipitate is formed at the bottom. It's not exactly brownish but yellowish brown type. Okay. Now we will add 1 milliliter of this concentrated H2SO4. Very carefully we have to handle it. It is 98% concentrated. It's strong H2SO4. Only 1 milliliter we will be taking. We will mixing into the BOD bottle, and you will see the precipitate will get dissolved. Everything will get dissolved in the concentrated H2SO4, right? And we will get more or less a uniform colorless mixture in the BOD bottle. And then from there we will take 100 to 150, or we can take 201 milliliter of sample from the BOD bottle, right? We will add starch as indicator. It will turn bluish in color or bluish green in color. Then we will titrate it against standard sodium thiosulfate solution of 0.025 ml. You have to be very careful while adding concentrated H2SO4, because if it falls on your clothes or on your hand, it would be very dangerous. Okay, now you see concentrated H2SO4 1 milliliter will be added into this BOD sample, and everything will get dissolved. देखिए कितना सुंदर सा जो रिएक्शन हो रहा है ना, everything is getting dissolved. This is wonders of environmental chemistry. How beautiful it looks, right? Everything is getting dissolved. A clear liquid layer will be formed. Everything will get dissolved. ये देखिए dissolve हो रहा है धीरे धीरे. We'll give a little bit of motion. थोड़ा सा इसको shake कर देंगे, ठीक है ना, so that proper mixing takes place. Very carefully you have to shake it, और देखिएगा एकदम जो है dissolve हो जाएगा. देखिए सर केयरफुली जो है इसको जो है थोड़ा सा शेक करेंगे, because concentrated H2SO4. This is the shaking, and everything will get mixed up, and a clear bottle you will see where clear transparent color has been developed. Okay, a little bit time it will take. It's a time taking process. DO determination, it's a time taking process. आप देखिए धीरे धीरे सब जो घुल रहा है. ओके, एवरीथिंग विल गेट डिसॉल्व. ओके, Now standard sodium thiosulfate solution 0.025 normality we will put into this burette. This is on, we will turn it off, otherwise liquid would fall. और हमने इसका हाइट भी बढ़ा दिया, सर इज शोइंग दैट इट इज क्लोज नाउ. ओके, standard sodium thiosulfate solution 0.025 normality. Now we will put it into the burette. We will note down the initial volume. इनिशियल जो है वॉल्यूम हम नोट डाउन कर लेंगे, ठीक है. अनफॉर्चुनेटली टोटल डिजॉल्व नहीं हो पाया, दैट इज बाय येलोइश टाइप ऑफ शेड यू आर सीइंग, बट नॉर्मली येलोइश शेड नहीं होता है, ठीक है ना. इट बिकम्स मोर और ट्रांसपेरेंट इन कलर. वी आर टेकिंग गिवन क्वांटिटी से 100 मिलीलीटर ऑफ the sample from the BOD bottle into this. Around 100 milliliter we have taken. We will add 1 milliliter of starch indicator, 1 to 2 drops. Since it is not totally transparent, that is why you will see that effectively blue color may not be developed. मैंने आपको बताया था ना, BOD वाटर्स जो हम BOD बोतल से लेते हैं उसमें जब स्टार्च ऐड करते हैं वी गेट अ ब्लूइश कलर, बट सिंस इट इज येलोइश इन कलर नाउ एंड दैट इज व्हाई it might not develop bluish color exactly, but bluish green color will be developed. Anyhow, that will also work. Now we will add starch as indicator. Okay, and it will turn, suppose, bluish green in color. We will add starch as indicator. Previously volume method was being done. Now starch indicator is being added. It might turn bluish green. Okay. See, color will start changing. See, color is changing. Okay. It is bluish green in color. It should be actually blue in color, but since our sample did not properly mix, so it has turned, suppose, bluish green in color. Now sodium thiosulfate initial level we are noting down. Initial level of sodium thiosulfate we should note down. Then we will start the titration process and we will give mild movements to the flask. Okay. See, the titration started, we are giving mild rotations. See, colorless, it has turned colorless. It has, within a few drops after having mixed, it has turned colorless. We will note down the final volume. We will note down the final volume. Okay. Then what is the quantity of sodium thiosulfate that is consumed? Final volume minus initial volume, that will give us the quantity of sodium thiosulfate. Then we will find out first the DO, dissolved oxygen. How we will find out? By ml of titrant Na2H2O3. कितना ml कंज्यूम हुआ है, that is final volume minus initial volume, multiplied by normality of sodium thiosulfate, that is 0.025, multiplied by 8, which is equivalent weight of oxygen. We will multiply it by 1000 to give the value of equivalent weight in mg per liter. Then we will divide it by volume of sample taken, say 150 ml or 100 ml of sample taken for titration. Okay, that will give us the initial DO level. Then we will store this BOD bottle with 300 ml sample in incubator, and after 5 days, after having kept it at temperature of 20 degrees centigrade, we will again take out the BOD bottle and we will determine the final DO by the exactly same process that we have discussed in this lab, that we have seen in this video. Exactly same process. Then we will find out the BOD as initial DO minus final DO. Okay, multiplied by dilution ratio, that is volume of diluted sample by volume of undiluted sample. That will give us the BOD in mg per liter. Okay. So students, calculation. So initially we will determine the DO. Initial DO we will determine, then we will keep it in incubator for 5 days at 20 degrees centigrade. Then we will find out the final volume, the final DO. So DO is obtained as ml of titrant, that is Na2H2O3. कितना ml use किया into normality of sodium thiosulfate, that is 0.025 into 8, which is equivalent weight of oxygen, into 1000, so that equivalent weight is written in mg per liter, divided by volume of water sample. Okay, volume of water sample taken for titration for in the flask. Okay, say 150 mg 150 milliliter is taken, or suppose 201 milliliter is taken, like that. Now BOD 5 at 20 degrees centigrade, it is obtained as DO consumed, that is initial DO minus final DO, okay, into the dilution ratio. Volume of dilution ratio is what? Ratio of volume of diluted sample by volume of undiluted sample. For example, suppose in the 300 ml bottle we have taken 50 ml of water sample, and we have diluted it to 300 milliliter level by distilled water. So what will be the dilution ratio? Volume of diluted sample, that is 300, divided by volume of undiluted sample, that is 50. This will give us dilution ratio as 6, and DO consumed will be initial DO that we find out by the lab procedure minus final DO that we will find out after having incubated it for 5 days at 20 degrees centigrade. Okay. So then we will go for the calculation. Suppose initial DO, for example, I have given a tentative example that suppose initial DO is 8.2 mg per liter. Suppose final DO is 4.4 mg per liter, right? And suppose sample taken is 50 milliliter. So BOD will be expressed as 8.2 minus 4.4, that is initial DO minus final DO, and initial DO will always be higher than final DO. Why? Because the DO depletes, DO decreases with time, because it is consumed for oxidation of organic matter by the microorganisms. Okay, so initial DO minus final DO, 8.2 minus 4.4. okay, so initial DO minus final DO into the dilution ratio. What is the dilution ratio? Volume of diluted sample, suppose 300 ml, divided volume of undiluted sample, that is 50. So we will be getting 22.8 mg per liter of BOD, and it indicates value is more than 6 or more than 5, so it is quite, means it is not suitable for drinking purpose unless there is no other option. Okay, so now conclusion. Here you see general guidelines for BOD values for classification of raw untreated water. There are suppose two classes, A and B. So drinking water source without conventional treatment but with chlorination, यानी हमने जो है बहुत ज़्यादा वाटर ट्रीटमेंट नहीं दिया है, लेकिन क्लोरिनेशन किया, डिसइंफेक्शन किया. ओके, सो व्हाट शुड बी द BOD वैल्यू? BOD वैल्यू शुड बी 2 or less. Actually for drinking water we prefer if it is less than 1, or if it is 1 to 2 or 2 to 3, like that, not more than that, otherwise it will be indicated as polluted. Then for another water sample with conventional treatment, दूसरा water sample इसमें treatment वगैरह दिया है ठीक से, BOD values 3 or less is expected. Note that it could cause problem in treatment, larger chloride demand and residual taste or odor problem might be generated. So since the test is mainly a bioassay, so I have told you students the environmental significance, we determine BOD so that we can judge whether the water sample is polluted or non-polluted. If the BOD value is high, that means polluted. If the BOD value is low, that means it is non-polluted, or it is rather treated. So since the test is mainly a bioassay procedure, it is necessary to provide a standard conditions of temperature, nutrient supply, pH, adequate population of microorganisms and absence of microbial growth inhibiting substances. The low solubility of oxygen in water necessitates the strong wastage to be diluted to ensure that the demand does not increase the available oxygen. And the 5-day BOD, basically I have told you in the theory portion also that it accounts for 68% of the BOD. We determine 5-day 20 degree centigrade BOD because it accounts for 68% of the CBOD or carbonaceous BOD. Okay, it indicates how much polluted is the wastewater or water. Okay, so that's all for today. needless to say that the session will be followed by Google form questions. You need to solve 4 to 5 questions by 11:30 PM tonight. So thank you so much everyone. Bye and take care. Thank you so much.